Swayam Prabha. Digital India. Educated India.
But in the endonucleases, many of them that we discussed were very specific in the sense. So, endonucleases, we discussed that they are, most of them are very specific. Specific in the sense, they recognize a specific basis. For example, if you remember, go back to your class where we said that DNA is made up of different bases and I am representing each of those bases by a specific alphabet there, right? A, T, G, C, and the hydrogen bond with their complementary base. So we said that your DNA recognizes specific bases and it cleaves off your DNA. Cleaves off in the sense, what is the reaction that is happening there? Hydrolysis of the phosphodiester bond. So it hydrolyzes the phosphodiester bond and it basically cleaves off those DNA, and then you have a large DNA strand, right? A, T, G, G, okay, C, T, C, A, something like this, right? And then you have a complementary base. So for example, T, A, and the endonuclease binds and then it does a phosphodiester bond cleavage and it actually cleaves off this big DNA strand into two different DNA strands. And you could do it at two different places so that you can remove any specific bases that you don't want and then you can religate them. Religate in the sense you can form again a phosphodiester bond and they'll form another DNA strand by removing those few bases. So suppose you know that few bases are creating any issue or that is causing any diseases, that is causing any problem, you can specifically remove those bases. But the problem is you need to have a specific recognition sequence because every enzyme can only recognize a specific recognition sequence. Only if you have those recognition sequences, it will be able to cleave that. But then we just discussed that it will be much more easier, it will be more nice if instead of the enzyme recognizing individual sequences, right? If we have another guide molecule where we can allow this molecule to recognize a DNA and you have a non-specific endonuclease that can cleave off your DNA, right? So this will be more useful than this because here for every sequence you need to have a different enzyme. So you need to have a large number of enzyme complexes because there are many bases. There are just 4 bases A, T, G, C. But the combination in which they can arrange is, if you remember human genome, we talked about 3.2 billion base pairs, right? So very, very large. So you cannot have so many enzymes to recognize different bases. But instead of that, if we have a strategy where we can have a guide molecule which will go and recognize the DNA, and then you have a non-specific endonuclease which will cleave off this DNA, which is your phosphodiester bond hydrolysis, then this will be more useful. But in the last class, we were tumbling upon few different things, right? So this is something I told you, right? So what did I tell you? Every protein is coded by 3 bases, right? If you remember. So if you see this, so you see these are all the bases. If you see, for example, this particular protein amino acid called as leucine. Leucine is coded by UUA, UUG, CUU, CUC. All these 6 bases can go code for one single amino acid. So based on the kind of amino acid you have, this is called as codon table. Codon table in the sense, you remember? So this is something that we discussed where you have DNA. DNA is transcribed into RNA and RNA is recognized by an enzyme called ribosome, and that is actually, that translates 3 bases. It recognizes 3 bases and converts them into one amino acid, and then that amino acid keeps on joining and then that forms a huge chain of amino acids or polyamino acids that will fold to form what is called as protein. So as you can see, each amino acid, some of those amino acids are being coded by only 2. For example, this particular amino acid, right here, phenylalanine. Phenylalanine is just coded by 2 sets of bases, whereas here leucine is coded by 6 different sets of bases. And somebody asked last time, right, does the number of codes per amino acid, does it depend on how frequently we see that amino acid? So it is not that easy to understand that because there is no strong correlation. For example, many of the bases that you see, so basically many of these amino acids that you see, do not correlate with the number of pairs or number of codons that individual each of those. For example, you might think here leucine has 6 different codons, right? It doesn't mean that there is more leucine content in our proteins than any of the phenylalanine. So it doesn't go that way, because this is still a process of evolution. We really don't know why one amino acid is coded by so many bases, whereas some other amino acid is coded by only 1 or 2 bases. So this is something that you should know, that this is called as codon table, where how bases are converted into amino acids. That means how codons are converted into amino acids, and amino acids keep on joining to form a protein. Yes.
Sir, does the basis that an amino acid is essential or non-essential, does that affect the number of codons?
No. So none of those things actually, whether it is an essential amino acid or non-essential amino acid. Only thing is there are 20 natural amino acids. 20 natural amino acids are coded by these bases, but there is no code. These are all called correlative studies. That means you see something, you try to correlate with something, but none of these correlations are actually possible. Yes.
So, niche base coding for an amino acid unique for an amino acid?
No. If you look at this table, I don't know. Shall I zoom this? So, for example, you see this part, right? It need not be. For example, you have there UAU, right? U, that is U, and whereas you see here it is UAU here. Okay, UAU, and then you have a UAA. That is coding for a tyrosine, and this is coding for a stop codon.
Yes, sir. But then for tyrosine, all the possible all the possible bases will always start with U, right? That's what I'm asking.
Oh, you are saying that? Okay. So yeah, for most, for all, if you are talking about a single amino acid, the starting of that will be the same, but it doesn't. The reverse of it is not true. In the sense, if something has C, that doesn't mean it will always code for tyrosine. Yeah, for one amino acid, the starting base would always be constant. Okay. So before we go into the molecular scissors in detail, CRISPR we start. Let us look at some information about proteins, because some of you are familiar with proteins, some of you are not familiar. Let's see what actually proteins are. As I told you, so you have an amino acid. Everybody recognizes the biomolecule amino acid here. So you have amino acid, and what will happen? So you have an NH2 group, you have a carbon, you have some alkyl chain group, then COOH, and then you have a H, right? And then you have another amino acid, NH2, and then you have C. This is R2, R1, and then you have COOH, and then you have a H. So this C double bond OH, C double bond OH, and this is what? NH2H, H. So this will result in loss of water, right? This will result in a loss of water, and this C double bond O will bond with this nitrogen. This is called as peptide bond. So when an amino group of one amino acid, and this is called a carboxylic acid, right? So when you have a carboxylic acid and amino, when they react, that forms a, this will form C double bond O NH. So basically what happened, you have linked two amino acids with the help of a single bond, which is called as peptide bond. Now when these multiple peptide bonds keep on joining, for example, this is an example, right? So when these multiple peptide bonds keep on joining, they form a large chain of amino acids. So those are called as polypeptides. I'm still not calling it a protein. I'm calling polypeptide. This is one peptide bond, then you have multiple of them forming, right? Those are called as polypeptides. And in polypeptides, this R, which is your alkyl chain, this could keep on, there are these 20 different amino acids that are possible. Now once this polypeptide forms, it will start taking a structure. Now why will it start taking a structure? You have a polyprotein, right? You have a polypeptide. Now this polypeptide, suppose it is in a water, right? Most of your, the cell is made up of what? What is the kind of environment it is in? It is all aqueous environment, polar environment, water. Now you see there are some amino acids like this, alanine, valine, leucine, isoleucine. These are all hydrophobic. Why they are hydrophobic? You see there are this alkyl chain, CH2, CH3, right? These are all carbon based, right? So these are hydrophobic. They don't like to stay with the water. Or you see there are some other amino acids like phenylalanine. These are also having aromatic compounds, right? So they don't like, you put it in the water, they don't like to have in the water. So what will happen? There will be a repulsion. So what it will do? finally it will try to minimize the energy. When there is repulsive interactions, the energy increases. So in the nature that you should remember that whenever there is a structure formation, whenever there is a conversion of one thing to other, it always happens to minimize your energy, because you have one amino acid and then that is your alkyl chain, and that is trying to interact with water, which is an unfavorable interaction because that is a polar environment. Here it is a non-polar environment. Polar and non-polar do not go well. So it tries to minimize your energy, and then it will try to fold. So it tries to minimize the energy and it will try to take a different shape. Okay, there are two common shapes that you find in proteins. So these polypeptides take shapes. One is called this beta sheet, where you see this. Okay, so you see this CNH C double bond. So you have a CNH C double bond O NH. So this, as I said, this forms a hydrogen bond, the C double bond O and NH. So you have a NH and then you have a C double bond O, right? And then you have continuously that. This will try to interact and form a hydrogen bond between them. Now, once these interactions are there, it will try to minimize the energy. Previously, it was in an extended form where there is a higher energy, and then slowly it will try to minimize the energy by increasing the interactions. And these interactions will lead to formation of a shape or a structure. So these are called as secondary structures, which is your beta sheet or alpha helix. Depending on the kind of amino acids and other things, it could take different shapes. So beta sheets or alpha helices. So it takes these two common shapes, and then once the polypeptide takes these shapes, it will further form many of these structures and folds to form what is called as a protein. So till now I have been calling it as a polypeptide, and a polypeptide takes different structures which are called as, to minimize the energy, and then they will form what is called as proteins. Now many of these proteins or many domains of these proteins can come together, and this is called as quaternary structure. This is called as primary structure, secondary structure, tertiary structure and quaternary structure, and many of the enzymes that you see in your body are actually these quaternary structures. Let me see if I can play this video. So initially, to start with, it was a linear polypeptide chain. It was in the high, this is, suppose you assume the energy as a funnel, right? To start with, you see, suppose this, the red mark you are seeing is corresponding to the energy state of it. Now you have an unfolded polypeptide chain, or you have a polypeptide chain which is interacting with its surroundings. Now, as I told you, the interactions can be very unfavorable because you have a polar molecule and a non-polar molecule. So interactions can be very unfavorable. So the energy is very, very high. So this polypeptide will try to locate all possible, it will try to locate a possible structure where it could minimize the energy. Minimize the energy since it can form a nice alpha helix or a beta sheet, those kind of secondary structures, and it can fold in such a way so that it can come to a lower energy state. You see, it looks for different structures and sees energy, and then once it goes into a minimal energy state, it takes different structures and it goes into the low minimal energy state, it stays there. Okay, so this is the minimal energy state, and then in that state the protein is in a folded form. Okay, once it reaches there, it forms a nice protein. It forms a alpha helical structure and beta sheet structure depending on the kind of amino acids and kind of interactions. Yes.
Sir, how exactly does it escape the local minima and go to the global minima?
Very nice question. You should look at what am I calling it as local minima and local. Okay. Now suppose you have a energy surface like this, right? This is an unfolded state where the energy is very high. Then it could have, this is the funnel. So this is the lowest energy structure. Now it could have on the way, it could have multiple local minima, right? It could get trapped in this also, right? Because it has to cross a barrier to come out of it. But these local minima are very low energy minima. So under normal conditions, you have a temperature bath, right? It is under a specific temperature. The temperature, at any temperature, it has an energy of kBT. If you know the Boltzmann constant, into its temperature. That is always the energy that is your temperature can provide. So if this, the barrier is lower than that, it could easily cross over that. Okay. Now many of these structures can actually result in this. So if you have a large, right, if you have a local minima that is energetically lower, then if suppose a protein gets trapped, or if protein gets trapped here, the energy from this T can help and it can easily cross it. Now there are always cases where this particular thing can be larger than this energy. So where this energy provided it will not be enough to cross this barrier. That could also be possible, right? Which not, but most of the cases these energy barriers are very small, so that it can cross by temperature. But there are always chances where it can actually get trapped in a non-minimalistic form. This is a minimal energy form. This is a local minima, but not a global minima, right? So this is called as misfolded state. Now this is called as folded state. That means this is the global minima, this is the lowest energy that it can take, and it can nicely stay there forever. But these local minima are the most dangerous ones because they can form a misfolded state. That means the protein will not form a structure that it was supposed to form, but it will take some other structure and it will stay there. And that is called as a misfolded state. And many of the diseases that you see are because a protein gets trapped in this misfolded state. For example, how many of you have heard of a disease called Alzheimer's? Right? So you all have heard. So Alzheimer's, why does it form? So you have there is a peptide at a molecular level. Any disease, when you say it has a molecular cause, there is a reason for it, right? So this amyloid, instead of going into its local global minima, it gets trapped in a local minima structure called amyloid beta. Okay, now that structure has a tendency to start aggregating because it is not the local minima, it is not the favorable structure. Now this starts aggregating and that will form plaques. Plaques means it will start forming these fibrils and aggregates, and that is how you get diseases. For example, many of the neurodegenerate diseases like Alzheimer's, Parkinson's, okay, even prion based diseases, all these many neurodegenerative diseases or other diseases are a result of this particular state. So if a protein doesn't fold into its correct structure, then some of them they will become dysfunctional. They will not be any more functional. Or the other problem is if they are not functional, that is okay. They are not doing their work. That is fine. If somebody is not doing the work, that is still okay. The problem is if somebody is not doing the work and disturbing the work of others. That is the problem, right? So if it goes into the misfolded state, it is not doing its work because it is not in the folded state, but it will start aggregating because the interactions are not very favorable. So it will start aggregating and forming other kinds of structures which will form large plaques, and that will cause many disease conditions. So this is a very, very dangerous phenomena, and it actually happens quite often, more than that. But I'm not going to talk about this in detail, but our body has a machinery to recognize if a protein is misfolded or folded. If it is a misfolded, it will try to degrade it. Okay, but even if that machinery fails, then it will start aggregating, it will form diseases. Yes.
So could you say this causes one type of cancer or something like that?
Cancer is a different, the problem with cancer is not a single term. We call it a single, but there are many kinds of cancer, and the reason for every cancer is the reason for a prostate cancer could be very different than a reason for a brain cancer. So that's the cancer is a very general term we use, but if you actually go into technical details of it, every cancer has a different origin. So it is not like just like your cough. Right? You are having cough. A cough could be because of millions of reasons. There is no single reason why somebody is coughing, right? So similarly, cancer is a symptom. You have an uncontrolled growth, but the reason for it could be multiple. Okay. Yes, there are also few cancers which are caused because of this misfolded states that are formed. Yes. Yes.
Sir, is it possible for the protein to have a combination of the helical structure as well as the sheet structure?
This is an example. This is a helical. The back side you see there is a sheet.
So what's the example? What's the name of that protein?
This is called ubiquitin. But there are many proteins. For example, even you take this protein, this is some other enzyme. This is a beta sheet. You see? This is a alpha helix. So there are actually there are more enzymes that are actually combination of it, because the energy minimization happens at a local level. So if suppose this 10 amino acids try to minimize energy, they could form a beta sheet, but there could be other amino acids that would try to form an alpha helix, but these are all part of a single chain. So they will form a protein consisting of all the possible combinations. But it is always not true that all of the regions will be folded. For example, you see this is an unfolded region, right? This doesn't have any structure. So it could be that there are folded regions, unfolded regions, and alpha helices, beta sheets, and also unfolded regions in the same protein. Okay, and these unfolded regions will give you the flexibility of the protein. Yes.
So let us say that a protein is now in its misfolded state. Wouldn't it be possible to heat, like increase the temperature?
We are talking about our body, right? You don't want to sit on 90 degrees.
Separately, like let's say in a lab, would it be possible to heat the protein to like escape that energy, to give that energy to cross the energy barrier and back into the folded state without it decomposing?
Good question. So let us take example. You all know, most of you probably eat egg. What is egg? What is the white surrounding of your egg? You have a yolk and then you have your white. It's not calcium. I'm not talking about the shell. When you boil an egg, albumin, right? It is nothing but a protein. So that is a protein that has been formed because albumin protein, if you actually break a raw egg, that is in a liquid form, right? You have a yolk and then you have a liquid form that is in a native form, but you have heated it, you have boiled it you have kept it in the boiling water. So that is a heat induced aggregation. That is actually an aggregate of your albumin protein. So it is not always true that when you supply heat, they will form individual proteins. Heat can also cause other kinds of interactions. In this case, it is actually forming an aggregate. But in principle, so as I said, body also has various mechanisms to recognize these misfolded proteins. For example, in our lab, we work on these enzymes called protein quality control. Protein quality control, just like you have a industry, right? You have a company. In company, there is a production. And then there is a quality control. Quality control means you have produced something, but somebody has to check whether everything is correct or not, because they are doing it in bulk, right? But you cannot be sure that everything is perfect. So you need to check what is called quality control. So there are these enzymes called protein quality control enzymes that recognize these misfolded proteins and unfold and degrade them. But it could also be happen that every process has a minuscule chance that it will escape all the possible proofreading mechanism. So it will escape that and it will start forming these aggregates, and it will result in many of the diseases. So many of the diseases, especially these neurodegenerative diseases, are because of this protein misfolding itself. Okay. Now we talked about this enzymes, right? So enzymes are nothing but proteins that come together and form a large complexes, and large part of these enzymes are nothing but proteins. Now these enzymes, are biocatalysts, right? They could do a catalysis of a reaction. For example, here it is a ATP, adenine triphosphate, right? It is doing a hydrolysis of this into your ADP and phosphate. So an enzyme can do a chemical reaction, or the enzyme can also do a physical reaction. Physical reaction means these enzymes can also act as motors. Motors in the sense they convert one form of energy to the other form of energy. For example, your piezos, right? So they convert one form of energy to other form of energy. For example, here because we have told this, right? It is important. Now, all these small peptides have aggregated. So when they form a misfolded state, they have formed this aggregate. Now there are multiple chemical reactions, and all those chemical reactions are exactly the same. So in these cases, it is not just possible to do a single chemical reaction to abandon all this. Now you have to physically pull each of these strands out so that it comes out of it. It's more of a physical process than a chemical process. So enzymes not only do chemical reactions, but there are also enzymes that do physical reactions. So in these cases, the enzymes are converting your chemical energy into mechanical energy, and they are trying to pull out these individual. If you have a large protein aggregate, they are trying to pull out these individual aggregates from that. Okay, so you should remember that enzymes are not just corresponding to your chemical reaction. They do a lot of physical reactions as well, and they apply a lot of mechanical energy as well. Okay, now this is some basic introduction about your proteins. Now let's go back to what we were doing. We were doing about molecular scissors, because molecular scissors involves both nucleic acids, that is your DNA, as well as your protein. So we have got a basic introduction of that, and let us see how these molecular scissors work. Now, so we discussed that these are your enzymes, which are proteins, and they recognize a specific endonucleases, that means specific bases, and they will cleave them off. And then what you really want is something to be non-specific, and then it goes there and then it cleaves off. Now this is done by a specific system called as CRISPR-Cas9 system. We just briefly talked about it in the last class. So it gives CRISPR-Cas9 system, and we said that it's a very, very novel technique and a new discovery that happened, that won Nobel Prize in just 2000, and it has revolutionized our understanding of gene editing. Okay. Now, so some of you are interested in science, some of you are interested in technology, some of you in medicine, different. But you should know how things work in science. So it's not like, scientists don't dream of something and suddenly they start doing the experiment, right? So there is a process that is involved. You all know. So basically, how penicillin was discovered? Do you know how penicillin was discovered? How was it discovered? Accidental discovery. So there is an accidental discovery, there is a systematic discovery. For you to be a good scientist, it is very, very important to have a good observation power. And I'll tell you a story here, how this CRISPR-Cas9 was discovered for the first next few minutes. And you will see how a system that is completely unrelated to gene thing has been transformed into something that does editing. Okay. So the process of this CRISPR-Cas9, so next 5 minutes is going to be about a story of how this was discovered. Okay, you can look at the names. Okay, names are important of the scientists, but let us look at the story. As I told you, so these people in end of 1980s, they were working on this bacterial DNA. They were trying to isolate the DNA, and they were trying to see what are the sequences that does this DNA have. Okay, sequences in the sense your AT, GC, all those kind of. So they are trying to see, because during that time the genome sequencing was still not there. Genome sequencing means today you can give your DNA and get your entire DNA sequenced, and you can know what height you will be, what is the color of you, till what are the hereditary diseases that you have. Everything can be known. But 1980s the genome sequencing was not there. So they were trying to understand how the DNA of different bacteria is. And they have, as I told you in the last class, they have accidentally discovered this particular thing. So what is interesting about this is generally you don't have many repeats in this, right? So, but what they have discovered is, now there is a specific bacteria. Let's not get into the details of it, but if you look at even E. coli, or if you look into Streptococcus pyonis or other kind of bacteria, what they discovered is, now you remember the railway track, what we were talking about the railway track, basically. So they discovered that there is some palindromic. So A, T, G, C. Okay, so there is a palindromic sequence, and then in between the palindromic sequence, you have some other DNA. Okay, and this is not a constant DNA, but you have a different DNA. And then again you have this palindromic sequence. Now suppose you are a scientist and you are doing this experiment and you find this DNA. What would you be more interested in? Would you be more interested in the palindromic part or the non-palindromic part? Why would you be interested in non-palindromic part? Because you are seeing something very unusual, because you don't find a lot of palindromes, right? There is a palindromic sequence, and this palindromic sequence is repeating itself quite often, which is unusual normally. So you are seeing something that is unusual. These are some other bases. These are some bases, and you will find these bases quite often. But the interesting part is you are seeing these things which are palindromic and they are repeating. So as if I were a scientist, because I am not as smart as you, so I would be more interested in these things, right? Because a palindromic sequence and it is repeating itself, that means it must have some use, right? But then, I think probably they were very smart, and they looked at it and didn't find any use for this. After that, what they recognized was this non-repetitive sequence, this blue part and orange part, this non-repetitive sequence is not doing anything in your E. coli or your bacteria. That means it is not coding for any proteins, it is not doing any function. But what they discovered is this particular non-repeating part is coming from a foreign DNA. Foreign means, so it's not, the DNA doesn't belong to this organism, but some other organism, for example, virus or some other organism. So this belongs to that DNA. and they discovered that beyond this, there are other genes that actually code for a specific enzymes called helicases and nucleases. So they discovered this. Okay, there is a palindromic shift, and then there is a non-palindromic shift, and they went on to that, and then they discovered that there are these genes beyond this that code for these two enzymes, helicases and nucleases. Do you know, we discussed what are nucleases, right? What are nucleases? What are nucleases? The enzymes that break the phosphodiester bond. So as I told you, the nomenclature is ACE. Any ACE is something that hydrolyzes. Nucleases hydrolyze nucleic acids, and there are helicases. Okay, what are helicases? Exactly. So you have a double-stranded DNA. Now it actually unwinds the helix structure of that and makes into a single strand. It unwinds the DNA. So these people discovered that there are three things that they have discovered. There is something that is very repetitive, and then there is some kind of interspersed sequences which are not part of your bacterial DNA. It is coding some other DNA, and it is not doing anything in the bacteria. And then there are genes beyond it which actually code for these two, because in a genome, when you are looking at a gene and when there is anything that codes for the enzymes, that means these enzymes will help in doing something for this part. Okay. Generally, if something is very close by, that means these enzymes this is called loci. In the same loci, if these enzymes coding for these enzymes are there, this will try to manipulate these things. So that is what has been observed, and then they started understanding this. So basically, this is the story that came up. So when a bacteria is there, so bacteria is an organism, right? So bacteria can also be infected, right? Bacteria can be infected by what? It could be infected by virus, right? There is a very famous virus called bacteriophage, right? Which infects the bacteria. So if suppose a virus actually infects a bacteria, and you all know that virus is really a non-living thing. So it's somewhere between living and non-living, because it actually doesn't have the components to do all these functions. What it does is it just introduces its genetic material like DNA or RNA into the host cell. For example, if it comes into our body, it will introduce DNA or RNA into our body and starts doing it takes control of our cell. In principle, it is like, what you see in science fiction movies. Virus by itself doesn't have any life. If you keep a virus here, it cannot do anything because it is not having life. But once it starts infecting, it will take control of our cells. Right? For example, you take HIV, if you take a COVID, right? All these virus particles takes control of our cells. Now this bacteria is infected by the virus, and the virus actually introduces its genetic material. Suppose this bacteria survives this attack. In the sense, the bacteria also has its own enzymes. Suppose it survives these attacks by cleaving of this DNA. What it does is, it incorporates. So this is the DNA of your virus. It incorporates that particular DNA into this CRISPR. We talked about this, CRISPR, cluster regularly interspaced, palindromic reports. So this is the original CRISPR, and now a new virus has attacked, and the bacteria has survived this attack. So now it will introduce that whatever the DNA that virus has given it, it will introduce it into this. Now why is it important? Why do you think this storing of it is like, memory card, right? So it will have all the information. So suppose if you look at this particular genome, if you look at this particular gene, it will give you the information of the generations of what all viruses have, so for example, this is not coming from just this generation. This has been passed on from many generations. So all the information that this bacteria has been infected with, it has all the virus information there. Okay, this is more like a storage, right? Vaccine or an immunity, yes. So the principle is same, where you have some kind of antibodies that are stored, but here we are talking about the DNA that is stored in this case, right?
Sir, how exactly does it come and attach itself?
It will not come and attach. So it will basically, the DNA of your genetic material of DNA of your virus is there. Now suppose that there is a lot of genetic material virus enters, right? Once it survives that, it will basically attach in the sense it forms this phosphor. There are these enzymes called ligases, right? We talked about that briefly. Ligases, which forms this phosphodiester bond. So it will form the phosphodiester bond and it will incorporate or integrate with the already existing DNA. Now this is like your bacterial immunity. Now next time the same, and this is called adaptive. Next time suppose a similar class of virus comes, what happens? So this, okay, before that, this DNA is transcribed into RNA and it forms a pre-CRISPR RNA. And then the enzyme actually cleaves off this into endonucleases, will cleave off this and it will store into multiple components. Now you could think this is a DNA and this is your RNA, and then beyond this, if you remember, beyond this there are these enzymes that code for this helicases and nucleases. So those will come and this forms a single complex, which is basically you have a DNA from the virus and then you have this is RNA which is converted from the DNA of the virus, and then you have a palindromic repeats, and then these are the enzymes that are coding for your helicases and nuclease. So all these are stored as a complex. So next time when a new virus of the same material comes or the same class comes and it introduces its own genetic material, because it already has this information on the DNA, right? So it goes there, recognizes that particular DNA or from the virus, and then you have these enzymes, nucleases and your helicases. The helicases will unwind the DNA and then the nucleases will cut the DNA, and so the new bacterial DNA that has been introduced will basically, it cannot do anything because it has cleaved off this DNA into individual fragments. So it is like your bacterial immunity. Bacteria has developed a way of immunizing itself because it doesn't have a vaccine. Bacteria cannot take a vaccine, right? So but it needs to have some kind of memory. That means this is the information that has been passed over generation, like from a mother bacteria to a son bacteria, although those terms do not matter. So you have a bacteria whose information is being passed from generations to generations, and it will store all kinds of virus that has attacked it. So if the same virus attacks again, so this will go there, it will recognize that, because why will it recognize that? Now it is storing it in the form of a complementary RNA. So as you remember, all this works on this complementarity. So it will go there, complementarily it will bind to this, and this nuclease enzymes will break those and it will form into two separate fragments, so that this viral DNA or viral RNA cannot propagate itself more, and this virus will not be able to affect anything for the bacterial growth. So this was discovered. This is called as bacterial immunity. Now how is this being used for gene therapy? Now one thing that you should remember is, you could ask me, there is a CRISPR DNA, right? I said it is transcribed into RNA and this complex is stored in the form of RNA. Why do you think it has to convert from DNA to RNA and store it in the form of RNA? Why can't it just store it as DNA, right? Can somebody answer that? And then I'll come back to your question. It is converting the DNA into RNA, and that RNA is binding it to its complexes and it is storing it. But why do you think it can just store it in the form of this DNA? Because this DNA also has the information, right? It is just processing the information. You have originally some information, it is processing that information. But it could store it in this form, but it will not do that. It will convert the DNA into RNA, and RNA will be stored along with this protein complex, so that whenever a new virus comes, this RNA will go and bind to the DNA of it and then cleave it off. What advantage do you think RNA will give compared to DNA, bacterial DNA itself? It will unwind the bacterial DNA itself. So that is one reason where you have a DNA attached to that, then that helicase will keep on unwinding the DNA. The other part is better or easier explanation is, you have a DNA, you have a RNA. Which one is more reactive? RNA is more reactive. DNA is a very stable structure. Now you have a new virus entering it. You don't want to have this entire process of unwinding it and then making it two separate strands and then this strand going and doing it, right? If it is already stored in the form of RNA, then because we know that this RNA has nothing to do with the bacterial part, right? It is not doing any function in the bacteria. So there is no RNA that corresponds to this that will come and bind to this. So this is a ready to use form, right? You have this ready, and whenever a new virus comes, it will just unwind, because already the DNA virus, DNA from the virus you have to unwind. If you also have to unwind your DNA, this is going to be a very slow process. So instead of that, you store it and keep it like your training. It's like your military. You don't want to learn how to shoot a gun after somebody attacks you, right? You should be ready with all your capabilities, and then when somebody attacks, you are ready to shoot them. Otherwise, it's not that I'm going to learn the shooting after somebody attacks me. That is of no use. So similarly, it is storing it in a very reactive form and not a stable form. So stability is not always useful. Sometimes reactivity is very important. In this case, reactivity is important than stability. It will go there and then it will just kill that new virus. Okay. Now this is your bacterial immunity. Yes.
So the virus DNA, when it is stored, it is not stored in the palindromic format, right?
It is stored in between. These black ones are your palindromic forms.
Sir, so what does a palindromic form do?
So palindromic forms are useful. For example, now you have this enzyme that is there, right? So enzyme should be able to recognize these things. There should be something that is a recognizable form, because the enzyme cannot recognize this part because this is always coming from outside. But you should have something that can be recognized by the enzyme, right? So it should have some, and the bacteria came up with the way that your enzyme can repeat. So if something is in a palindromic form, then that means it is corresponding to your immune system in that case. So your enzyme can easily recognize that and form this complex and then immediately go and do that, right? It also needs to have that recognition.
Sir, also once the bacteria gets this immunity, won't the virus, like after some time, won't it just mutate and change its DNA?
Yes, it could. So again, it depends on, as I said, it's not like one or two bases. There are a large number of bases. For example, 26 bases, 30 bases, right? Now, if one or two bases are mutated, then even then it can nicely recognize that. But if the entire thing is mutated, then if entire 26 are mutated or maybe 15 are mutated, then it will obviously not be able to recognize. But if one or two bases are mutated, it can still be able to recognize that. Okay. Yes.
Sir, is it possible for it to misrecognize the DNA?
When there, as I said, this is not a 0 and 1. Like, it's not a binary. There's always, so for example, there are 10 bases, and then there are, even if suppose a new DNA comes which has 8 bases which are similar to this. So DNA is not going to, these are multiple, if there are 10 bases and then you have a complementary strand of 10 bases. So there are 10 different interactions, right? That means there is some energy associated with that. Now, if there are 10 bases of which 8 actually match with that, you still are going to have 8 different interactions. So energetically it will be less favorable, but it still can form. So these are all based on, it's not like 1, 0. These are all interactions that are there, right? So it could still form a complex. Now this is basically the bacterial defense system that you saw. But how was it used to do a gene editing is something that we are going to look at it. So as I told, these are the two pioneers, Jennifer Doudna and Emmanuel Charpentier, who thought that why not we use this, because bacteria has different ways of defending itself. Why not we use this bacterial defense system to be useful? That is something that can be used in our day-to-day life, or that can be useful for mankind. So basically these are two different scientists. You see, one person is good at observation. There are few people who observe that these are the things that happen. Then comes the people who thought, okay, this is an observation, this is a thing that is happening in nature. How can we use it for the use of a mankind, or can we use it mankind, or the in the sense humankind? Can we use it for the benefit of your humankind? And these two people did those experiments where they have used this particular, because this particular DNA, when they did the experiments with just this complex, it was not forming any kind, it was not showing any activity. So they discovered that there is another piece of DNA that is missing, that is called as nucleic acid, that is called tracer RNA, which will bind to the palindromic repeat. So which has a complementarity to the palindromic repeat. So it forms a complex with the palindromic repeat, and this is stored as your individual molecular scissors. So depending on, so suppose this is your test DNA, some DNA that you want to break, or some, and so you make sure that this is all constant. This red one and the black one is constant, because this black one is recognized by the enzyme, and the red one is important because it is complementary to the black part, which is a palindromic part. So depending on the sequence that you have, you can always modify this sequence, and this will go and bind to that. Once it binds, okay, So once it binds, it actually cleaves off those and completely cleaves a particular DNA, and then you have a activity of nucleases. Although I am showing it as a simple cartoon, there are many structures that are available. These are all very complex enzymes, and this is the structure of the enzyme complex that we talked about. As you can see, that's why I gave you the introduction of your alpha helices and beta sheets. So these are not some cartoons, these are actually amino acids that are folded into a specific shape. So once it forms, it cleaves off that particular DNA, and then so that it can it can no longer be it is no longer functional. It is completely broken out. So I'm just giving you one example here. Okay, so now I have a DNA. This is some DNA sequence that is coding for a certain protein. Now I want to basically remove this part because that is causing some disease. I want to remove that part. Now I have to design a CRISPR RNA sequence so that it can nicely go and bind to that. What is the first step? The first step is you basically design a complementary sequence to that. So this is the complementary sequence. As I said, complementary sequence means A for T, G for C. Okay, you design a complementary sequence and then you convert that sequence into RNA. How do you convert the RNA? You convert your T into U. So this green part, which is the complementary of the part that you wanted to mutate, you convert into, do a complementary part, and the complementary part you convert into RNA, and this is all constant. This is all same for a particular enzyme. Now once you have this enzyme will bind to, this is your Cas9 enzyme, that will bind to this part. Okay, once it binds, so you can actually cleave off this two particular strands, and those two strands are separated out, and you will get a you will basically lose it. Now the question is, what if you want to add new bases to that? You can also add new bases by, for example, you can take some. So this part is gone now. This entire part is gone. You can take new bases that are complementary to this and complementary to this, and whatever the new bases are there, you can add this, and this will form a phosphodiester bond, and you can have new bases. So you can remove few bases, you can add new bases, and you can completely modify the DNA to get whatever the DNA that you wanted. Okay, so let me just give you few. So here is what I showed you. So you cut that DNA, and then you do what is called as PCR, where you actually identify few bases that are complementary to this part, and they'll keep on adding, and then you can add new bases to that. So I'll just tell you applications of this is all done in a test tube, but at the end of the day you want to transfer it into humans or higher organisms, right? So that part of transferring this test tube work into humans was done by these two scientists called Feng Zhang and George Church. So you can see the applications of this. So this was used in identifying your detection of your CRISPR, detection of your COVID, yeah. So your coronavirus basically during the COVID, okay. And then this was also, it has multiple uses. It has already been used to improve the efficiency of the crops. It has been used to increase the, it has been used as a therapeutics. So if you talk about therapeutics, the common therapeutics are either you do a sanitation, vaccine, surgery, or medicine. These are the commonly accepted therapeutics. But the next one is genome editing, in the sense, like for example, you have some disease, you can remove. For now, you cure the disease. You take the disease and then you try to cure it either by giving medicine, or you undergo a surgery and remove that part, or you give a vaccine, that is prevention, or you maintain the sanitation so that you will not even get that. But the next part is, even by looking at that disease, you can actually remove the genetic part of it so that it will not get transmitted further and further. So that can be done, which is your next, maybe once you guys grow up, probably around 10 years from now, this might become this is actually a reality now. And this is something that people are looking into. Yes.
So does this mean that we could genetically engineer ourselves to become immune to diseases, and that would pass on, right? So you could engineer the entire human race to be immune to a certain disease?
Yeah, there are two parts to the question. At this level, for example, once you are a mature human, you have all your proteins already there, right? So already those particular proteins, for example, I told you about a CCR5 gene that codes for a protein, CCR5. Now you already have that protein, right? So it is, you cannot remove that, but you could remove it in the next generation. That means you could remove that gene, and that could be passed on so that the gene does. So you can do it at an embryonic level. But this is possible, as somebody asked last time, this is possible technically. But ethically, nobody will allow you to do that. You cannot design your own, in the sense, you cannot have a next generation of kids to have, you know, a disease free, or that is not allowed ethically, that then everybody is going to be intelligent, everybody is going to be disease free, everybody is going to look alike, like everybody is going to be smart, right? So that is, your entire heterogeneity is lost. So that is not used, but only it is used when you have a specific very inherited diseases that are very you know, somebody is having an inherited disease that is being propagated again and again, and that is a death causing disease, then ethically it could be done, but still now the ethics don't allow that. It is a possibility, but technically it is a possibility, but ethically it is not possible. Yes.
So, but how does elimination of something like disease in, you know, like at an embryonic stage reduce heterogeneity in any way? Your ethnic markers and everything still remain, right?
Sorry, repeat that question.
How does elimination of something like disease at like an embryo stage in any way affect the heterogeneity of the
No,
species?
so disease, it's okay. But you said, can we design ourselves?
Yes.
That is so you cannot, so then, so whether somebody accept or not, there are many traits that are genetically inherited. Now, if suppose you have some kind of, you know, good looking eyes or good hair, you want to engineer it further and further, right? So then basically you are eliminating a specific kind of hairstyle, or you are eliminating a specific kind of eye thing, right? So you are losing the heterogeneity part in that. So this way you are basically, it's like, you know, you go to a market, you have a product of exactly same type. Now, if suppose this thing comes, you are basically manufacturing somebody of exactly same type, right? You are going to, because you know that all the phenotypes that you are seeing are cause of your genetic. So it's basically, you will have exactly same kind of looking, maybe some minor things, but all of them will be looking similar. All of them will be having the same kind of intelligence level. All of them will be having or not having same kind of diseases. So basically, you are removing the entire heterogeneity. This is not a product, right? Humans are not products. So that is okay for a manufacturing unit. So that is why ethically it is not allowed. Okay, so as I told you, this has been used, and there is a lot of research that is going on. Basically, there are many diseases that are being addressed. I'll not talk about many diseases here, but you know, there are specific genes that have been identified, and you could modify those. But you know, there are some unusual applications as well. For example, people have genetically engineered pups. Okay, Dalmatian pups, so that Dalmatians are more prone to some genetic disorders that causes gallbladder stones in them. Okay, so they have genetically engineered pups that would actually do not have that, or they have actually engineered this one. Where is that? Okay, the mosquitoes. Yeah, they have actually people have engineered mosquitoes where because mosquito is also a vector, right? It will bind to a specific parasite, and it will. Now we are killing the mosquito, but we know that the real cause is not the mosquito, but it's basically the parasite that is sitting in there. So now they have engineered mosquitoes which cannot bind to those parasites, so that the mosquitoes will no longer be able to, they are no longer vectors for this transmission of diseases. So that has been engineered, and there are many other things that have been used. And one example I told you as in the first slide itself, right? People have actually, this is not a science, this is not a myth, or this is not a science fiction. People are actually, scientists in the US are trying to, regenerate. So basically they got some DNA of the woolly mammoth, right? And then that's very close to elephant's DNA, but there is some changes. So they are removing, they are taking elephant's DNA and they're comparing with that, and they're trying to introduce whatever the additional genes are needed for your woolly mammoth, and they're trying to introduce that, so that you actually have a woolly mammoth that is basically a species that has been extinct few millions of years ago, that is being regenerated. Again, as I said, these are all technical possibilities, but people will not, the ethical reasons, it will not be easy. But still, you have your CRISPR, which is going to find its ways in having a reduced hunger in the sense, you can devise crops that can produce a better quality, like, you know, a better quality crops, or you can have plants that can give more oil, or you could have inherited disease free. All these are possible with CRISPR. There are many things that are possible technically, but there are some things that are not allowed ethically. Okay, so this is something that I want to end with, where you have a technique which has been discovered in just a decade back in 2010, which is kind of revolutionizing the entire way we look at your genome, and you can actually modify things and do changes at the genetic level. So if you are more interested, just Google about CRISPR-Cas9 system and there is a lot of information that is available.